Okay, the next thing we're going to talk about so this has to do with kind of the basic introductory stuff. The first section of the class is going to be really about the phylogenetic perspective. What is the phylogenetic perspective? How do you think about it? I mean, this is really very different than most microbiologists. So when you think about microbes, how do you divide them into groups? Some people divide them based on stains, gram-positive, gram-negative. Some people divide them based on what they do for a living, phototrophs versus heterotrophs, anaerobes versus aerobes. The phylogenetic perspective argues that you ought to think about them in terms of how they're related to each other genetically. In the same way that when you think about animals, for example, you divide them into phyla and so forth and that way, rather than putting all herbivores together in a group and all carnivores together in a group, etc. We're going to do that for bacteria. So let's start with microbial diversity generally. This is actually a pretty tough question. How are microbes similar and different from each other? And how do we measure that? Now, this is an old wood cutting from Anthony van Leeuwenhoek, and it's pretty amazing. I mean, does this look like your typical sort of bacteria that you think about? Probably not. Most microbiologists at your level, when they think about a new organism, will ask the question, is it a rod or a coccus? That's kind of the beginning point, right? That's morphological diversity. And so it certainly is true that bacteria are different or similar to each other based on their shape. Cocci are easy, they're more or less spherical. Rods are cocci where there's a plane and a cylinder in between the two ends, right? Usually. It turns out that if you look at a microscope slide of bacteria, a lot of them are those shapes, but a lot of them are other shapes as well. And the diversity is probably way beyond what you think of. This thing, Halo quadraticum, is shaped exactly like a bathroom tile. It's square, it's flat, it has sharp 90-degree corners. This is in salt lakes. It's an archeon. These are gas vacuoles inside. When it divides down the middle, like so. And the next division plane is perpendicular to that. And the next one is back to the original plane and back and forth. And so it creates sheets of little tiles. Is that typical? Is that what you think of when you think of the shape of bacteria? Probably not. They also come in filaments. They're triangles. A relative of this is three-sided. They look like Doritos. A lot of organisms change shape depending on growth conditions. Every year in the agar-degrader lab experiment, I have a student who struggles continuously to get a pure culture, but he can never separate that big coccus from the long, thin rods. And the reason they can't separate them is that those two organisms are one and the same. When the cells go into stationary phase, they circularize, they form cocci. But when they're actively growing, they're filaments. Even E. coli. An experienced person can look at microscopically at a culture of E. coli and tell you whether it's logarithmically growing or stationary phase E. coli, because the cells are quite a lot smaller when in stationary phase. They almost look like cocci. They can also be in different cellular arrangements. And so if you have two identical looking rods, you can tell a culture apart based on whether the cells are mostly individual, whether cells are in pairs, whether they're in long chains, whether those chains are branched or not. Sometimes cells lay side by side, like a picket fence. All of those things are clues to the identity of the organism, by the way, and how they divide. Rod-shaped cells that divide down the middle tend to form chains or pairs. Cells that divide lengthwise tend to form picket fence palisade arrangements. And so it tells you something about the physiology of the organisms. This is a film. This is a biomat. This actually is from a salt turn in Mexico, where they've taken just a core through this microbial mat. And then here's a picture of it. This is 2 millimeters right here. Look at the complexity of organisms in that. This is a physical arrangement of thousands of species of bacteria and archaea, arranged in a very specific order where every layer has a function in that community. This is a tissue. It functions exactly like the tissues in your body. The difference is it's a tissue made up of a bunch of different species instead of one species where the cells are all different forms, which is the way it is in your body, right? Parts of these are anaerobic, parts are oxidizing sulfur, et cetera, et cetera, et cetera. Size. Typical bacterium. How big is a typical bacterium without reading it off the screen? A couple of microns, right? More or less. Some are smaller, some are bigger. Here's a bacterium. This thing is Ecological phylogenetic. It's found in the guts of saltwater herbivorous fish of the tang family. Those are the yellow ones you see in saltwater fish tanks a lot. This particular one comes from the brown surgeonfish, a relative. This is paramecium. This thing is 700 microns long. It's big enough to see with the naked eye. It is a gram-positive bacterium related to Clostridium. That's not the biggest bacterium there is. Thiomergita is about like so. Single cells. For years, this thing was thought to be a protist, and no one to this day can cultivate it in the lab. Structural diversity. When you think of structural diversity in bacteria, the first usual thing is, well, does it have a gram-positive envelope or a gram-negative envelope? Turns out most bacteria have a gram-negative envelope, meaning that there is an inner membrane and an outer membrane, both made of phospholipids and proteins. Other envelopes are unusual, but they do occur. Some organisms have a single envelope layer and a cell wall outside of that. In some cases, they have an extra membrane, but not made of phospholipid, made of mycolic acid instead. Many organisms have protein cell walls. This is Coarceum cryptophyllum, and the cell wall is made of protein, and these proteins are arranged in a crystalline array on the surface. An S layer. S layers are actually quite common in the bacterial world. When you look at a diagram of a typical gram-positive or gram-negative envelope, you're looking at E. coli and Bacillus subtilis and almost no other organism. There is a huge diversity of details of cell envelope structure. Never assume that because an organism is gram-negative or gram-positive that you know what its envelope structure looks like. Organisms have flagella, fimbriae, holdfast stalks, buds, capsules, all kinds of surface and structural stuff. They have internal structures. Spores are an obvious one. Daughter cells. What do I mean by that? Epiphyseum does this. Here's the mother cell. These two things inside are going to be the offspring. The offspring form inside the original cell. The mother cell lyses and releases the offspring. Again, this is an organism related to Clostridium. The process of developing those daughter cells is homologous to the endospore forming genetic machinery. The internal structure of bacteria is just as complicated as eukaryotic internal cell structure. Forget the days of drawing a bag and a bunch of enzymes inside doing stuff. Bacteria are not like that. The fact that we don't think about it much is just because we can't see it under a microscope, but that doesn't mean it isn't there. Enzymes and structures are highly organized in bacterial cells. Otherwise, they don't work. Those of you who've had metabolic regulation have seen this, for example, in chromosome structure. Metabolic diversity, though, is the really big one, I think. Eukaryotes are boring metabolically. They're heterotrophs. They take in organics and they oxidize them in some way or other. That's it. Actually, to be perfectly clear, they grow only by substrate phosphorylation. You breathe oxygen, right? But it's not you. It's the mitochondria. Mitochondria are bacteria. They're alpha proteobacteria. What about plants? Plants don't photosynthesize. The chloroplasts do. Chloroplasts are blue-green algae. Permanent endosymbiotic bacteria. The nuclear cytoplasmic part of a eukaryote, the lineage that really makes up eukaryotes, they're all just substrate level phosphorylizing metabolically. They're boring. Ecological diversity. Here's a hot spring in Yellowstone. This is Moose Pool. pH 3, more or less. 85 degrees centigrade. Teeming with life. It's like a giant sulfolobus fermenter. If there's liquid water on Earth, there almost certainly is something living in it. There are only a few weird environments that we know of where there's liquid water on this planet and not something living in it. There are some springs in New Zealand where the pH is above 13, where there's no evidence of life. Hot springs in the seafloor where the hydrostatic pressure of the overlying ocean increases the boiling point really high. Above about 160 degrees, there's no evidence for life. Some of those springs, the water comes out at 650 degrees centigrade, and there's no good evidence for life in that. But otherwise, if there's water, there's life. From the deepest groundwater there is, little tiny strings of water in the cracks of rock, a mile below the surface, all the way to the water droplets that make up clouds. There's stuff living in that. From 10 degrees below zero to 150 degrees centigrade. From pH 0, Metallosphaera grows very nicely at pH 0. That's half molar sulfuric acid. All the way up to at least 12, 12 and a half or so. It turns out that alkali is a little bit harder than acid for most organisms, and there's good reason for that. Let's see. Symbiosis. I mentioned mitochondria and chloroplasts already. We live in a microbial world, in the microbial world, not separate from it. Behavioral diversity. Really, bacteria, behavior? Sure. Chemotaxis is a trivial example of this, right? If you put a drop of glucose in a tube, all the E. coli will swim to it. There's a great video somewhere, a guy at Indiana University, whose name escapes me at the moment, did it, where he's shined a spectrum, a rainbow, across a microscope slide and showed that rhodospirillum likes a certain wavelength. And he knew that because all the bacteria form a band right there at that wavelength on the slide. This is chlorobium, and this heterotrophic beta proteobacterium is a cell in the middle, a single rod-shaped cell in the middle. These two organisms can only grow together, in nature at least. The chlorobium is photosynthetic, but non-modal. The heterotroph is, well, heterotrophic, but modal. These organisms live together, and the chlorobium provides energy and fixed carbon to the heterotroph, and the heterotroph in return gives it motility. These things live in meromictic lakes. These are lakes near the ocean where there's a layer of saline water at the bottom and fresh water on top. And they have to find exactly the right part of the water column where there is both reduced sulfur and light. And the heterotroph can't measure light, but the phototroph can and signals that information to the heterotroph in terms of controlling its motility. These organisms, chromosomes and cells divide in synchrony. They are very tightly coupled to each other. If that's not behavior, I don't know what is. Developmental cycles. Here's Streptomyces. This is a common soil bacterium. It behaves a lot like fungus because it has the same ecological niche. Filamentous branch filaments. Some phases, the filaments are individual cells, but mostly they're syncytial. That is, the nuclei flow freely in the cytoplasm, no boundaries. All of the red cells are dead here. These are aerial hyphae. These are spores. Very complicated life cycle. If you look at a colony on a plate, it's quite common, for example, to see concentric rings, right? What's that mean? That means the colony grew in spurts, sometimes diurnal cycles, right? You get a ring every day. Well, if you look at a bread mold, you can tell how old the mold is by how many rings it has, because they grow in a diurnal cycle. Here's Myxococcus xanthus. That's this stuff over here. And here, this is a colony of E. coli. And the Myxococcus, having been released from its cysts, is swarming over the E. coli colony, lysing and eating the E. coli. And doing so, notice, I mean, you can't see individual cells in here. These are waves of the predator as it surges over the E. coli colony. So where does all this diversity come from? And how do you measure it? This is science, right? You have to measure it. All of this kind of diversity is based on genetic diversity. The genes, the chromosomes that make up these organisms, and how they control the form, the behavior, and the metabolism of the organism. We can sequence genomes now. Most of the information in that genome is still beyond our capability of understanding, but we do understand some of it, and so we can learn a lot about an organism by sequencing its genome. You can then take genes within that genome and use it to determine how related different organisms are, quantitatively, and use that information to create phylogenetic trees of organisms, and that's what this is. We'll spend the first couple of weeks talking about these trees, how to make them, and what they mean. This evolutionary diversity, phylogenetic diversity, is really the key to understanding all the other kinds of diversity. And that's what we'll be talking about this semester. Any questions about this so far? I'm going to save this again.